Another special type of animal virus or human virus is HIV, human immunodeficiency virus. It's a type of retrovirus, which we haven't talked about yet. And this is a group of RNA viruses that have a very unique replication process because they can do reverse transcription. Now, just to begin with, I just want to say what HIV is. HIV is a prevalent virus, and it's a virus that infects and eventually kills white blood cells. White blood cells are basically our immunity. So as the count of white blood cells goes down, you lose your immunity over time, and you can develop something called AIDS, which is acquired immunodeficiency syndrome. Okay, so HIV, it's an RNA virus. It's a retrovirus. It means that it has RNA as its genetic material, but in addition to having RNA, it has an enzyme called reverse transcriptase. So we haven't talked about this yet, but some viruses inside their capsid, in addition to having their genetic material can code for enzymes, and they can have enzymes that do special things like reverse transcription. So HIV, again, is an RNA virus. It's surrounded by a protein coat, a capsid. It has an envelope. Envelopes are made of a lipid bilayer. And the way that HIV works is it primarily infects white blood cells. By the way, we call those CD4 plus T cells because you will be learning about immunology soon. And this white blood cell is very important for your immune system's response to infections. So the way that this works, and I have an image here, we have a picture of HIV virus infecting a white blood cell is in retroviruses. So again, not there's other examples of retroviruses. It's not just HIV, but when we say something is a retrovirus is an RNA virus that can do reverse transcription. And the reason it can do reverse transcription is because it has an enzyme called reverse transcriptase. Reverse transcriptase is an enzyme that can convert RNA to DNA. And the reason why this is significant for a virus such as HIV is HIV is an RNA virus. So when it infects white blood cells, it will inject its RNA, but then using its enzyme reverse transcriptase, it will convert that RNA to DNA. And this is very important for the virus because then it can take that HIV viral DNA and integrate it into the human's own DNA. And this is not good. So this is why HIV is one reason why it's a pretty significant infection to get. So let's go through the life cycle of HIV. And the nice thing about all of these is whether we're talking about bacteriophage or DNA viruses or RNA viruses or retroviruses, they all pretty much have a similar pattern of the virus has to attach, it has to inject its genetic material. The genetic material gets made into virus proteins. The virus particles get put together and then they leave the cell to go on and infect more cells. So let's go through how replication or copies of HIV are made. So HIV enters our body through bodily fluids, and that could be through dirty needles, contaminated needles, through sex, and it will bind to receptors on white blood cells, and the white blood cells are called CD4 cells, the specific type of white blood cells. And once inside the cell, the HIV will release its RNA and its reverse transcriptase, which is an enzyme that converts RNA into DNA. It does reverse transcription. And then this newly formed HIV viral DNA will get integrated into our own cell's DNA with the help of an enzyme called integrase. So they also have integrase. The host cell's machinery then will begin making HIV viral RNA and proteins, and then all those new viral proteins will get put together and they will bud out from the cell. And in this process, they will acquire an envelope, a lipid bilayer from our cell's membrane. And this process, unfortunately, also destroys the host cell, so the white blood cells. That's why you see the white blood cell go down over time. So HIV over time will deplete our body's white blood cells, weakening the immune system and leading to AIDS, acquired immunodeficiency syndrome. And this unfortunately leads to the body not being able to fight off infection and disease. Now, there is no cure for HIV, but antiviral therapy can control the virus, which can help people a lot because HIV is really a very complex retrovirus that weakens the immune system. And this reverse transcriptase from the retrovirus is what's responsible for converting the virus's RNA into DNA, which again gets integrated into our own DNA. And the thing that's important about this virus, HIV, is that it has a very high mutation rate. And that's because the enzyme, the reverse transcriptase, is prone to a lot of errors because it doesn't have proofreading ability. And this means that it doesn't correct mistakes that happen when it converts RNA to DNA. So there's a high mutation rate in HIV. So the errors made by this reverse transcriptase result in a lot of mistakes. That's why there's so much diversity of the virus strains. And this constant change is pretty bad medically. The reason why it's bad is because it's been so hard to create an effective vaccine because the vaccine has to stimulate an immune response that recognizes all or most of the virus strains or variants. And if you keep having these variants, it's really hard to create a vaccine. So in order to just control the HIV, to control the rapid mutation of it, people who have HIV usually take a lot of drugs, and we call that a combination of drugs, a cocktail of drugs. This therapy usually has reverse transcriptase inhibitors, which block this enzyme from working, and this prevents the virus from replicating, which is a good thing. There's other inhibitors in the cocktail which can stop different stages of the replication cycle. So that's why it's important to understand replication cycles. And then just as an example, there is a drug that's very popular called AZT. It's a type of reverse transcriptase inhibitor. Specifically, it's an analog, or it looks like one of the bases. It looks like thymine. And what it does is it mimics this, and when it mimics this, it basically stops continuous transcription of the virus. And so you stop production of new viruses. So this drug cocktail is very, very important for people who do have an HIV infection. Okay, next we're going to talk about coronaviruses. Coronaviruses are a very large family of RNA viruses that can cause illnesses that can range from the common cold to more severe diseases. There's different strains of coronaviruses. They're enveloped viruses, meaning they have an envelope, they have a lipid bilayer, and they have spikes on their surface. They look like crowns. That's where they got their name, coronaviruses. SARS-CoV-2 is the coronavirus strain that's responsible for COVID, which is the pandemic illness that began in 2019. The SARS in SARS-CoV-2 stands for severe acute respiratory syndrome because it causes serious respiratory disease. And then when we think about the transmission of SARS-CoV-2, it's primarily transmitted through respiratory droplets when someone coughs or sneezes. And the virus can also be spread by touching surfaces contaminated with the virus and then touching your face. So touching your respiratory illnesses. Okay, so here's more information on it. COVID symptoms can be mild, they can be severe, and the way to control it is through vaccines, and we'll talk about that. But talking more about the virus originated in bats and they were transmitted to animals. When we go from one species to another, this is called jumping species. So these strains had a very, very high mutation rate to be able to go from animal to human. Usually that's not a very simple thing to do. And it was a new strain for us at the time. That's why it caused a pandemic, because people's immune systems was not used to seeing this. So we had nothing to protect us from it. Okay, and then just a little bit about the origin of coronavirus. It was discovered in 2019, and we were seeing people to have pneumonia-like symptoms in China. We now know that it causes COVID, and I want everyone to leave class knowing that it's a specific coronavirus strain that causes COVID because there's many other strains, and we've had the other strains before. A lot of common colds are caused by other strains. The strain that causes COVID again is called SARS-CoV-2, severe acute respiratory syndrome coronavirus 2, and it jumped from animal to human because of mutations that happened. When we look at the virus structure of SARS-CoV-2, it is an RNA virus. It's an enveloped virus. It has spike proteins, and spike proteins help the virus attach to our cells. And it has a capsid, it has a membrane, it has an envelope. So that is the structure of coronaviruses. When we think about how it enters our cells, it uses its spike proteins to enter our cells. So the way that it uses its spike proteins is its spike proteins bind to specific receptors on our cells. One type of receptor that they can bind to is called the ACE2 receptor. The reason why this is important is because we see ACE2 receptors on our lungs, our kidneys, our gastrointestinal tracts. It's in our lungs, so that's why we were seeing a lot of like breathing problems that people had when they were getting diagnosed with SARS-CoV-2. And SARS-CoV-2 antibodies sometimes can stop this interaction, so this binding. Okay, treatment and vaccines. We now have multiple vaccines for SARS-CoV-2. There's mRNA vaccines, there's other types of vaccines. There are antiviral drugs too that we have, but they usually work when you take them at the beginning of an infection before there's a big viral load. And then another idea that we were doing during the pandemic that was a temporary band-aid is when there was no vaccine and there was nothing, and we actually do this with a lot of viral infections when we don't have treatment, is if you have someone who had an infection and then they were fine, there's always that outlier that gets it and nothing happens to them. What you do is you get a blood sample from them and you extract the antibodies, and then you can take those antibodies and you can inject them in someone who's sick, who's unable to fight the infection. So just different ways that we were able to tackle coronavirus. SARS-CoV-2, again, it's an RNA virus, it's an enveloped virus, and it has spikes. Okay, for our next topic, we're going to talk about, we just talked about viruses, and now we're going to talk about outcomes of viral infection. And sorry, these should say 1, 2, 3, 4, but anyway, you get the idea. So viral infections can result in different outcomes, and these can be acute, chronic, latent infections. And in some cases, viral infections can transform normal cells into cancer cells. So we're going to talk about all of these. We're going to go one by one, starting with acute infection. Okay, so in an acute infection is basically a type of infection where the virus will basically, you get the virus quickly. So you get it very quickly and you have a short duration of symptoms. And when I give you an example, this will make more sense. So the virus will enter the body. It will show symptoms quickly. Quickly can be days to weeks after the virus enters the body, and then you'll have an immune response, and then the infection will go away. Examples of this, now this will make more sense, are things like the common cold or the flu. And basically what I'm trying to say is in these types of infections, like if someone has the flu and they're next to you, and they sneeze on you, you typically get this the infection within like 1 to 2 days, within days or weeks, and then it also goes away relatively quickly. Like you don't have the flu for like 6 months. So that's what an acute infection is. It's a quick onset of symptoms. You're sneezing, you're coughing. They last for a short time period and then they're gone. So that's an acute infection. That's the definition of an acute infection. Examples are flus or colds. Okay, unlike acute infection, there's something called a chronic infection. So a chronic infection or a persistent infection is when the virus continually replicates and is present in the body over a long time. So in an acute infection, it comes and it goes, it's gone. In a chronic infection, it's a persistent infection where you see virus present in someone, usually in low numbers, for a very long period of time. And the viral infections usually develop very slow and they last for a long time. And these can potentially result in chronic health issues and bad outcomes. So, an example of a persistent infection is hepatitis infections. So, specifically hepatitis B and C, not so much A, but I'll talk about hepatitis B and C. So, in hepatitis B and C, what you see is that the person gets this, and these are transmitted in somewhat similar ways to HIV. You don't see the symptoms for a while, they affect the liver, but once you see the symptoms, they're there for a very long time, slowly affecting the liver. So it's a chronic condition. Okay, next we have latent infection. So in latent infections, the latent infections are where the virus is in your body, but is dormant without causing continuous symptoms, but it can reactivate. So the best way to explain this is an example. An example of a latent infection is like a herpes infection, the herpes that causes cold sores. So here, when we think of cold sores, one thing we can think of is the virus that causes cold sores stays dormant in nerve cells, but it can reactivate at any time period due to stress, due to illness. So it'll reactivate multiple times in your lifetime. And when it's not reactivated, it's just laying dormant. So the virus remains asymptomatic. Asymptomatic means not symptomatic for long periods of time. And then suddenly you start seeing symptoms. So they come and they go, they come and they go. And when they're not there, that means the virus is just dormant. Another example of this is, for example, the virus that's responsible for chickenpox can enter like this latent state after initial infection, hiding in nerve cells. And then years later, it can reactivate as shingles, which is a very, very painful rash. So just keep that in mind. And very few people actually get this reactivation. It's about like 15%. Okay, there's more examples, and then we're going to talk about viruses that can cause cancer. Okay, one more thing about latent infection. So with latent infection, after your initial infection, the virus isn't detected, meaning you do not see symptoms unless it's reactivated. So the reactivation again can happen due to you getting sick, due to you being stressed, due to anything that decreases your immune system can trigger it. And so again, some examples of those were shingles, and this number is not 30% right now. The latest data, this is according to your textbook, but the latest data is 10 to 15%. Okay, now we get transformation into cancer cells. So some viruses can cause cancer. And so what cancer is first of all, we have to talk about what cancer is. Cancer is cells that keep dividing. They just keep dividing and dividing and dividing. And then you end up having a lot of cells, which we call a mass of cells, which we call a tumor. And so anything that can trigger cells to divide continuously, we say that that's oncogenic. So there are viruses that can trigger, so certain viruses can trigger changes that lead to normal cells dividing continuously. And so with these viruses, what they do is they basically trigger this in one way or the other. Examples are hepatitis B and C viruses may, it doesn't mean they will, but they may be able to trigger liver cancer. Human papillomavirus has a very high risk of causing cervical cancer and other types of cancer. EBV virus may be associated with lymphomas, which is cancer of the immune system, etc. So that's what it means. So any virus that can trigger cells to divide can cause cancer. And for our final slide on viruses, we're going to talk about plant viruses. So viruses also infect plants. They can cause diseases that can severely impact agriculture and also food. So understanding plant virus is very important so that we can develop resistant crop varieties and we can have sustainable food. So these are very similar to other viruses. They have the same way of replicating. The main thing that I want you to take from this slide is there's something called tobacco mosaic virus, and it infects tobacco and related plants. So whenever you see plants like this, those are infected with viruses. There is no cure for most viral plant diseases. So they're very important for agriculture. So just keep that in mind. You can't just get rid of the virus. There's no cure. You have to get rid of the plants. So that's why they try to work on breeding genetically engineered plants that are resistant to viruses. And now that we are done with viruses, we're going to end this lecture by talking about prions. Prions are not viruses at all. They have nothing to do with viruses, but I'm going to talk about them with this lecture. Prions are basically infectious proteins, and they cause really, really bad, devastating diseases like mad cow disease and other diseases. And what prions are is misfolded proteins, and they're unique in that they're misfolded proteins, and they can cause healthy proteins nearby to become misfolded. So they stand for proteinaceous infectious particles, and you can get them by eating contaminated meat, basically. They can be in transplants, organ transplants, surgical instruments. That's why you have to sterilize everything really well. And again, they're misfolded forms of a protein that's normally present in brain cells. So this misfolded protein is transmissible and it causes fatal, means deadly diseases. So let's talk about what happens with this, and we'll go through with it. Okay, so there's no nucleic acid, there's no DNA, there's no RNA. We're not talking about that. We're talking about proteins. So prions are misfolded versions of a normal protein called PRPC, which is cellular prion protein. When PRPSC comes into contact with normal proteins, it unfortunately causes them to misfold into more prions, and this creates a bad chain reaction. So this PRPSC binds to the PRPC on the cell surface and converts it to the bad prion, the scrapie protein. And this can accumulate in the brain and cause more misfolding. This can lead to cell damage and death and result in this spongy degeneration of brain tissue, which is what we see in prion diseases. So this is basically what you have. So you have the normal form of the prion, and then you can have the bad form of the prion of the protein, the misfolded, and it can react with the other proteins and turn them into the bad one. That's my simple version of trying to explain prions. And then when we think of prion diseases are fatal. They're basically deadly. It causes brain diseases, and these brain diseases creates holes in the brain that we call plaques, and they can be inherited, they can be transmissible. That's why it's very important to make sure that you're eating clean meat. And so we see brain diseases. The most popular one or common one is mad cow disease, which is bovine spongiform encephalopathy, and Creutzfeldt-Jakob disease. So there's Creutzfeldt-Jakob disease. And so again, with prion disease, it's basically demonstrating how misfolded proteins are very bad and they can be transmissible. And here's a good video of it.
In 1982, Stanley Prusiner discovered that a sheep disease called scrapie was caused by a proteinaceous infectious agent, an infectious agent composed entirely of protein without any nucleic acid. Prusiner named these new agents prions for proteinaceous infectious particles. Prions do not reproduce like other infectious agents by multiplying or growing. Instead, they cause other normal cellular proteins of the same type to misfold into the infectious form. For his work, Prusiner was awarded the Nobel Prize in Physiology or Medicine in 1997. Since 1982, several other diseases have been shown to be caused by prions, including bovine spongiform encephalopathy, also known as mad cow disease.
And then just one more, just showing you the diseases.
There are several human diseases known to be caused by prions. Creutzfeldt-Jakob disease is rare, occurring in about 1 out of 1 million people. It usually appears in midlife between ages 20 and 70. The average age of onset is about 50. Recently, a form of the disease called variant CJD, VCJD, has emerged. It was first detected in the United Kingdom in people who had eaten beef from cattle infected with bovine spongiform encephalopathy, so-called mad cow disease. Once the first symptoms are seen, both variants of CJD progress very rapidly from onset to disability to death. CJD affects the cerebrum, where it causes holes called vacuoles and amyloid plaques, clusters of prion proteins, and infected cells. Gerstmann-Straussler-Schenker syndrome, GSSS, is a rare human prion disease. GSSS is caused by a single mutation in the gene encoding the prion protein and is
Okay, so you get the idea. And then finally, here are the study questions for the lecture on viruses. And I hope you learned a lot about viruses.